Thanks everyone for joining us for the 3Rs Collaborative and IQ webinar series. Today's webinar will be on Multi Organ MPS. For those of you I haven't met before, my name is Lauren Young. I'm the program manager for the 3Rs Collaborative, and I'm excited to kick us off this morning with a quick disclaimer and then a brief introduction to the 3Rs Collaborative. So I'm just going to read right off the slide here. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So as I mentioned, I'll start with a quick introduction to the 3Rs Collaborative. So this webinar series is hosted through our MPS initiative, which aims to collaborate to accelerate adoption of MPS in scientific research. And we seek to increase adoption and regulatory acceptance of MPS in coordination with animal studies. We're able to do this through our diverse collaborations across the scientific field. So this is just our leadership and board of directors on the screen here. But as you can see, all members come from various different industries as well as institutions, and this makes us a strong force to enact our goals. Our MPS initiative includes over 50 member institutions with over 75 different members, all of those which you can see on the slide here. And we have 5 different aims within this initiative. So first, to provide thought leadership and expert consensus from developers in combination with stakeholders relevant to MPS. Facilitate appropriate discussion, collaboration, and sharing. Create direct engagement between developers and regulatory agencies on MPS, develop external partnerships and collaborations, as well as to create resources to facilitate increased engagement with MPS developers. And we're able to do this through our 3 different work streams. Our regulatory work stream, our technology expo, and our end user and education. And I'm just going to highlight what each of these 3 work streams are currently working on in 2025. So for our end user work stream, we are hosting quarterly MPS workshops, of course, one of which is happening today. So we've already hosted webinars this year on AI and machine learning, retina and ocular MPS. Of course, this one's on multi-organ MPS, and then we have a few upcoming at the end of this year. We're also presenting at various conferences like the MPS World Summit, continuing submissions to All-Text Corners, and we're looking forward to organizing some additional webinars on relevant topics for end users. So stay tuned for those. Any of our previous webinars that we've hosted in past years and this year, you can see on our presentation page. And then next is our regulatory group. So in 2025, they've published a publication on perspectives on using MPS for regulatory applications. As I mentioned, it is fully published, and the QR code links to that publication there. Again, promoting at various conferences like the MPS World Summit and WC13. They're also continuing forward progress on a cross-platform DILI project and beginning development of a benchmarking project for end users, which is really exciting. And then finally, we have our Technology Expo. So this launched in 2021, but as of 2025, we continue to add different disease areas and tissue filters, allowing end users to find the relevant MPS for them. continuing regular promotional posts, and then we're also nearly finished developing an exciting new addition, which is our publication hub, which will have filters to explore relevant publications by company. So we're hoping to launch that very, very soon. So definitely stay tuned for that as well. I just wanted to highlight that we have two final webinars for 2025. So definitely write these dates down in your calendar and keep them earmarked, and stay tuned for the topics for these webinars. For our webinar today, we will be using the Q&A function. Questions will be answered after each presenter, time permitting. If there isn't any time, we'll be hosting a roundtable at the very end where any remaining questions will be answered. So definitely put your questions in the Q&A. If the function doesn't work for some reason, feel free to also use the chat. If you have any additional questions about our initiative, becoming one of our 3RC sponsors, or the webinars in general, my email is on the screen there. And with that, I will hand it over to David Kukla from the IQ MPS initiative.
Thanks so much, Lauren. For those of you who don't know me, my name is David Kukla, and I just wanted to give a quick overview of the IQMPS before we get started with all the talks. So the IQ is a collaboration of pharma and biotech companies, and it was created as an affiliate within the International Consortium for Innovation and Quality in Pharmaceutical Development. And the IQ-MPS has four main aims. The first is to develop external partnerships and collaborations to help enhance the inclusion of industry priorities. Second, to serve as a thought leader for MPS developers and stakeholder organizations. Third, to provide a venue for appropriate cross-pharma collaboration and data sharing. And finally, to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS in industry. So the IQ is currently composed of 26 member companies, and within that, there are over 150 active participants, and there are representatives from drug safety, 3Rs, ADME, and PKPD. And here's our 2025 Executive Committee. Anya was our previous chair in 2024, but in 2025, Rhiannon David is our current chair, Kim Homan is the vice chair, and Philip Hewitt is our vice chair-elect. And here I'm just showing a breakdown of our IQMPS structure. At the top, the Executive Committee provides leadership and guidance in the management of IQMPS business and affairs and implements strategic plans that are recommended by our steering committee. And that being said, the steering committee is made up of 2 representatives from each of our 26 member companies, and they are the primary decision-making body for the IQMPS. Then within the IQ, we have 4 major work streams, and I'll kind of go over those on the next few slides. But then within each one of these work streams, there are several different subteams. So the first work stream is regulatory engagement, and their mission is to build regulatory confidence in MPS and align on a common understanding of how MPS data should be handled in regulatory submissions by sharing industry knowledge of MPS utilization with regulators and collaborating to overcome challenges utilizing MPS for drug development applications. The second work stream focuses on publications, and their mission is to outline the contemporary pharmaceutical industry perspective and considerations for developing, evaluating, and characterizing MPS models to support drug discovery and development. And then the pilot project workstream works to initiate near proof of concept studies for data sharing or prospective collaboration for qualifying MPS systems based on a specific context of use. And then last, the strategic partnerships and communications workstream. Their aim is to develop external partnerships and collaborations to help enhance inclusion of industry priorities by making the IQMPS the partner of choice for pharmaceutical MPS education, alignment, and engagement. And then additionally, the workstream disseminates IQMPS positions and learnings and influence external stakeholders. Also, they create a venue for bringing novel information into the affiliate and educate member companies. And then finally, they also support MPS adoption among the next generation of scientists. So I just kind of give a quick overview of the IQ, but if you have any questions or want to learn more about what we do, please feel free to check out our website, which I have linked here, as well as our LinkedIn page, or you could contact us directly at info@iqmps.org.
Cool. Right, so my name is Mornay. I'm from CN Bio, a senior computational scientist. And today I'll be talking about some work that we've been doing at estimating oral bioavailability using dual organ gut-liver MPS. So to start off with, bioavailability is a pretty important key parameter during lead optimization, and is very important when determining dosing, especially going into first-in patient studies. A higher bioavailability means you can have a smaller concentration or lower dosing and still hit this therapeutic window here. There are a couple of ways you can enhance your bioavailability by enhancing oral absorption or permeability of the drug, or reducing some first-pass metabolism effects. Now, in today's current toolbox, we have some limitations when it comes to determining bioavailability, especially for animal to human extrapolation. We don't have very good correlation with animal studies and observed human in vivo bioavailability estimations, and this becomes an issue when you're trying to narrow down on a reliable bioavailability estimation. We do have the KCO2 model, which is used for predicting absorbance. However, the KCO2 model does lack in some ways, where you don't have relevant metabolism of the drug in the KCO2 model due to low expression of some key enzymes. So what we've done at CN Bio is we've established a dual organ gut-liver MPS in collaboration with Altus to make this a fully primary cell model. So here you have your gut organ, which is connected to your liver organ via some microfluidic flow. This is a snapshot of the gut cells, the primary gut model from Altus RepliGut, just to compare that to the KCO2, which we've also tested in our system. You can see the differences between the two, but you also have this mucus layer that is produced by the RepliGut cells. Then the liver model, we have our standard CN Bio liver model, where you have the scaffold with holes in them that you see the cells onto. The cells attached to the scaffold form 3D tissue, and then the scaffold, the cells are perfused with media. Now, to profile bioavailability in our system, this is what a general assay would look like. On day 0, you would seed your primary jejunum cells. Day 9, seed your primary hepatocytes directly into the dual organ plate. On day 13, you can then transfer the jejunum cells into the dual organ plate. At this point, you have your dual organ model ready to be experimented on, to be tested. The model is viable for experiments for between 48 and 72 hours, in which case you can dose with your drug and analyze through LCMS. So just to show that we maintain some functionality of both of the organs throughout the course of the experiment, here we have some tear measurements of the gut barrier organ, where at the dotted lines you have this where the co-culture begins. We have maintained high tear measurements after that as well. And then for liver functionality, we also have maintained albumin production throughout the course of the co-culture, and the same for STIP3A4 expression as well. So I'm going to be going into a case study that we did with midazolam, just to show how we go from experiments to bioavailability prediction using this system. So we chose midazolam, because it is metabolized by CYP3A4, which is also present in the gut cells. So you do have some metabolism by the gut during first pass metabolism of midazolam. So here we have the bulk RNA-seq just to demonstrate that we have more expression of these relevant CYPs in the replicat model than in the KCO2, especially for the CYP3A4, which is also then shown by this qPCR data, where we do have more expression of CYP3A4 in the replicat compared to the KCO2. So this is what your the conditions would look like if you're trying to do this kind of experiment. We chose to do a liver only, gut only, and also a gut and liver experiment. The liver only, you only have liver cells, so no gut cells. The drug is dosed into the liver compartment, and this replicates your IV scenario. We then have a gut only experiment where you don't have any liver cells, you only have gut cells. Drug is dosed into the gut apical, and the reason for this experiment is so that you can piece apart the gut and liver metabolism of the compound. We then have the gut and liver condition, and we have both your gut and your liver cells dosed in the gut apical compartment, and this replicates your oral dosing of the compound. So to get into the data, this is what the liver-only data look like. You see you have a decrease in midazolam over time. It appears in your liver compartment. Oh, sorry. This is the hydroxymidazolam, which is created over time and then subsequently metabolized as well. We also did a cell-free control, and the reason for your cell-free control would be to determine if there's any non-specific binding of the compound during the experiment or any evaporation that you need to take into account. In this case, the concentration of midazolam remained pretty stable throughout the course of the experiment, and so we determined there was a negligible non-specific binding or evaporation. In our gut-only setup, we did this for both replicat and the KCO2. You can see the midazolam concentration decreases over time from the gut apical as it's absorbed into the liver compartment. This is the same for KCO and replicat. However, when looking at hydroxymidazolam, you can see that the KCO2 is not metabolizing midazolam at all, where the replicat is, and you do have production of hydroxymidazolam. So then for the gut-liver experiment, this is simulating our oral dosing of the drug. The drug is absorbed from the gut apical compartment. It then appears in the liver compartment. We get a peak concentration, after which it's further metabolized. The same happens for hydroxymidazolam. The peak appears at a later time. You do have hydroxymidazolam present in both the KCO and the replicat experiments. However, this is because of the presence of the liver cells in this experiment. Right, so once you have all of this experimental data, we then want to extract some key parameters such as the permeability, gut and liver clearance, from which we can get bioavailability prediction. So how we go about this is we first start off by constructing a couple of different mathematical models with the use of ODEs. Depending on certain assumptions that we might make, we'll generate a couple of these models and then fit them all to the experimental data. We can then rank these models according to how well they perform, how well they are able to describe the data, and then we'll select the best performing model for further analysis, where we can take a look into the parameter distributions as well as the confidence intervals, and then from that we get a bioavailability prediction. So this is an example of what our best performing model looked like. In this case, we were able to go with quite a simple model and still get pretty good results. So having more complexity in the model isn't always useful. So in this case, we could get only just two compartments with our combining the gut apical and enterocytes as one compartment, and then another compartment with the gut basolateral and the liver compartment. So this is what the model fit looks like with the data, able to describe the dynamics in the data quite well. From our model fit, we then use Bayesian inference to take a look at the parameter space. And the reason we do this is so that we don't just narrow down on a single value for our predictions of each of these parameters. We instead can report a range or a confidence interval of what we think these parameters might be. We can also see correlations between the parameters as well using this method too. So then from these parameter distributions, we can then narrow down on confidence intervals, and these confidence intervals or these distributions then filter into our later analyses where we do Monte Carlo simulations so that we can get or we can predict a range of what we think the bioavailability might be. So once we've extracted those parameters such as the permeability, gut clearance, and liver clearance, we can then move on to estimating bioavailability. The way we did this was separately identifying an FA, FG, and FH, the product of which gives you your bioavailability. FA is the fraction of the drug that is absorbed after taking a dose orally. FG is the fraction of the drug that escapes your gut metabolism into the portal vein, and then FH being the fraction of the drug that escapes your hepatic metabolism. So for FA, the way we did this was we took the apparent permeability value and then we scaled this to get an FA value. For KCO2, we had pretty high permeability, so we predicted an FA value close to 1. With our primary gut models, this is with the repligut, we got a lower permeability, so a lower FA value that was closer to that of the in-human in vivo predictions or observations. Moving on to FG. For FG, it was a bit difficult to scale the gut clearance value up in order to predict FG that way. So what we did was we opted to use model simulations where we calculated the area under the curve, a situation where the model was left unchanged, so it had gut metabolism, and then we simulated the model again by switching off gut metabolism. So we had no gut metabolism in one instance. We then get the ratio of the areas under the curve for our simulations with and without gut metabolism, and that ratio is then an indication of the FG. For the KCO2 MPS, we get an FG of pretty much 1 because there's no gut metabolism in that case. And with the primary gut model, because we did have some gut metabolism, we then get a reduction in the FG close to 0.8. However, this does fall outside of the range of in vivo observations, but it is an improvement on the KCO2 model. Then for FH, we calculated FH by scaling our CLINT, so the intrinsic clearance in the liver. We get similar values for both the KCO2 and the primary gut models, is because the liver component in these models is essentially the same. We have a bigger range here in the primary gut liver because you do have metabolism in the gut and your liver, so that increases some of the uncertainty in the observation here. However, both of these fall quite nicely in the range of the in vivo observed values. So then moving on to F, we get the product of FA, FG, FH, ending up with our oral bioavailability prediction for midazolam. In the primary gut model, we get a prediction that lies quite nicely within the range of the in vivo observed values. Each of these data points is a patient for which the bioavailability was determined. The KCO2 value is quite a bit higher. It doesn't really fall within the range, and we do get quite a nice improvement using the dual organ primary gut liver MPS that we used in the study. So just to summarize quickly, we show in the study that we can produce a gut liver model that is made up of only primary cells, and from this we can estimate, or we can get a prediction of the bioavailability as well as its components FA, FG, FH. We also show that the importance of using a correct gut model that has the relevant metabolism of the compound when determining F gene. And we show that, with mathematical modeling in combination with this MPS, we're able to provide a tool that can be used to identify promising drugs and predict a bioavailability prediction. So, thank you.
My name is Muya Lee. We appreciate the opportunity to present our pillar precision plate platforms designed to enhance 3D cell-based high-throughput screening. As we all know, traditional 2D cell monolayers and simple spheroid models fail to replicate human physiology, leading to costly failure in drug development. To address this need, Bioprinting Laboratories has developed and commercialized pillar precision plate platforms which can integrate 3D bioprinting, dynamic organoid culture, and high-throughput screening to ensure greater accuracy, scalability, and cost-effectiveness in predictive compound screening. So why our technology over various microphysiological systems already out there? Our platforms offer substantial advantages. Assay volume can be miniaturized 10 to 100-fold, increasing efficiencies and reducing reagent costs. Unlike other MPS systems, cells and spheroid loading on the pillar plate is exceptionally fast and straightforward, utilizing microarray 3D bioprinting and the manual stamping method. Dynamic cell culture enhanced organoid maturities, promoting more physiologically relevant results. Compatibility with the standard lab equipment eliminates the need for additional investment in new instrumentation. Operation is pump and tube-free, simplifying workflows and reducing potential system complexity. We have introduced and commercialized 3-pillar plate, 2 complementary perfusion plates, 1 deep well plate, and ranges of accessories to design to support both static and dynamic cultures of 3D cells and organoids. Let me briefly explain how it works. Cells encapsulated in hydrogel can be efficiently loaded on the pillar plate, which can then be sandwiched with the perfusion plate to facilitate dynamic organoid culture. The 144 pillar plate features 12 by 12 arrays of pillars, while 144 perfusion plates include a complementary 12 by 12 arrays of perfusion wells and 12 by 2 arrays of reservoir for cell culture media, which interconnected by microchannels to enable dynamic 3D cell culture. When the pillar perfusion plate is placed on a digital rocker, it generates forward and backward fluid flows. SolidWorks simulations and flow rate measurements indicate an average flow rate of 5 to 20 microliter per second, which can be adjusted by changing the tilting angle. These flow rates closely resemble those observed in microcapillaries, enhancing physiological relevance in dynamic organoid culture. Using SolidWorks, we simulated velocity profile underneath the pillar in the 144 perfusion plate. As you can see, the unique designs where pillars are sandwiched into the perfusion wells facilitate rapid mixing of cell culture media, effectively minimizing diffusion limitations for nutrients and oxygen for 3D cell culture. We have successfully demonstrated the cultivations of various organoids using our pillar perfusion plate technology, including liver tumor, brain, intestine, colorectal cancer organoids, and more recently, cardiac organoids. These findings have been published in high-impact journals such as Advanced Healthcare Material, Biofabrications, and Lab on a Chip. Let's briefly explore the unique features of our pillar perfusion plate platforms for human organoid culture. Traditionally, organoid culture has been performed using 6 and 24 well plates with the matrigel dome, ultra low attachment well plates, micro well plates, thinner flash, mini bioreactor, and perfusion plate. However, these platforms are primarily designed for large-scale static cell culture and often lack compatibility with the high-throughput screening. To address these limitations, we introduced a miniature high-throughput dynamic 3D cell culture system utilizing a pillar perfusion plate. So cell encapsulated in hydrogel can be efficiently loaded on the pillar plate using 4 simple methods, including manual dispensing with the 1 milliliter pipette tip, pillar plate stamping into the loading plate, spheroid transfer from ultra low attachment well plate, and cell printing by 3D bioprinters on the pillar plate. iPSCs and expandable organoids can be suspended in the hydrogel and uniformly printed on the pillar plate using a 3D bioprinter. You can see that we can load the mixture of the cells and hydrogels in the disposable tips, and then the machine can pick up these samples and then print it directly on the pillar plate in a high throughput manner. The typical coefficient of variation for cell printing ranges from 10 to 20%, demonstrating high reproducibility in cell loading by 3D bioprinting. These images showcase 3D cells, have 3D cells suspended in arginate and loaded on the pillar plate using simple pillar plate stamping method. The coefficient of variations remain below 15%, demonstrating high reproducibility in cell loading. Following cell encapsulation in hydrogel, the pillar plate can be seamlessly integrated with the Petri dish, deep well plate, or perfusion plate, enabling both static and dynamic cultures of 3D cells and organoids. And ensuring uniformity in 3D cell culture is essential for high throughput compound screening. Our study demonstrates that spheroid and organoid cultures on the pillar plate exhibit consistent size uniformity, enhancing reproducibility and assay reliability. Additionally, the pillar perfusion plate is fully compatible with the standard laboratory equipment for organoid analysis, eliminating the need for customers to invest in additional instrumentation. These images depict spheroid cultures in the ultra-low attachment well plate and the pillar plate, stained with the calcium AM and ethidium homodimer to distinguish live and dead cells. Cancer spheroid cultured in ultra-low attachment well plate exhibit significant cell death in the core, attributed to the formations of necrotic core. In contrast, cancer spheroid cultures in the pillar perfusion plate demonstrated minimal cell death in the core, benefiting from these dynamic 3D cell culture conditions. Furthermore, organoid maturity was significantly enhanced in the pillar perfusion plate due to miniaturization and rapid diffusions of molecules. Human liver organoid cultures on the pillar plate demonstrated significantly higher maturity compared to those grown in 24 well plate. Similarly, brain organoid maturity was further improved through dynamic culture in the pillar perfusion plate, highlighting the essential roles of rapid diffusions of signaling molecules in 3D cell culture. These findings are further validated by cryo-sectioned images of brain organoids cultured under static and dynamic conditions for 31 days on the pillar plate. The layer structures are notably more pronounced in the brain organoid cultured dynamically in the pillar perfusion plate, highlighting the impact of dynamic culture on organoid development. Organoid cultures on the pillar plate can be tested and imaged in situ without the need for harvestings or isolations of cells. crucial advantages for high-throughput compound screening. Whole-mount immunofluorescent imaging can be performed by simply attaching a thin glass slide to the bottom of the pillar plate, facilitating efficient image acquisitions. By utilizing expandable liver organoids printed on the pillar plate, we successfully achieved scale-up organoid production under both static and dynamic culture conditions. Human organoid cultures on the pillar plate were utilized to evaluate hepatotoxicity of model compounds and generate dose response curves, demonstrating their potential for predictive toxicology applications. Additionally, human organoid cultures on the pillar plate were utilized to evaluate liver regenerative capacity following drug-induced injury, a process traditionally assessed using animal models. Our findings demonstrated a significant elevations in inflammatory gene expressions post-injury, which subsequently decreases during expansions and recovery of the liver organoid, highlighting the organoid potentials for studying liver regeneration. Cryopreservation is essential for scalable human organoid productions and on-demand delivery to customers. Our pillar plate enable on-chip cryopreservations of human organoids, streamlining high-throughput organoid-based assays. Additionally, we evaluated the efficiency of virus infections in day 35 brain organoids using adeno-associated viruses carrying GFP genes under both static and dynamic conditions. The pillar perfusion plate demonstrated high efficiencies in organoid virus infections, highlighting its critical roles in gene editing for human organoids and disease modeling. We also evaluated immune cell-mediated killing of liver tumor cells using the pillar perfusion plate, where 3D cell Hep3B liver tumor spheroids were loaded on the pillar plate, and the NK92 cells, along with the immune cell activator berberine, were introduced in the perfusion plate. This setup facilitated immune cell infiltrations into the liver tumor spheroids, leading to effective immune cell-mediated cytotoxicity. Finally, multi-organoid interactions can be facilitated within the pillar perfusion plate by cultivating individual organoids on the pillar plate and rotating pillar plate at a 90-degree angle. This approach allows individual organoids to be maintained in a specific organoid differentiation media while enabling organoid interactions in co-culture media, supporting complex cellular communications and disease modeling. And if you are interested in our pillar perfusion plate products, you can request a lab demo or free samples at 3dbpl.com. Thank you.
Thank you so much for this opportunity. This is the first time that TheWell Bioscience is going to participate of one of these webinars, so thank you so much for the opportunity. I will be presenting how our biodirectional hydrogel system can support some MPS models. So let me just put this here. Firstly, I'd like to quickly introduce biodirectionals for those ones who might not know it just yet. So biodirectional system, it's a pure synthetic, xeno-free hydrogel. It's room temperature stable in its liquid at room temperature and retains its injectability properties after gelation for a long time, which makes it compatible with lab automation and high throughput applications. It has a really fast and easy protocol of only 20 minutes. It's pure transparent to ensure the users to have high resolution images. And it's really easy to harvest the cells after culturing. Also, it's synthetic, it's pure synthetic. It eliminates any problems of batch-to-batch inconsistencies. And also it makes it really easy for us to tune it to modify either the ligands or some properties as the mechanical strengths of the degradability properties of the hydrogel. We have two main groups of hydrogels. The blue gap, the ready-to-use versions are designed based on the cell type or the application. These are the easiest to use. Basically, the user just have to mix the hydrogel with the cell suspension and plating, and that will be it. And in the case of the high concentration hydrogels, it allows the user to control the whole composition of the of the matrix and the properties. So it comes with a dilution solution to allow to adjust the mechanical strength. And also with the high concentration versions, you have the ability to mix and match to create your own matrix. Just so you know, our aim is to be able to provide an end-to-end solution to support the whole study. So we don't only have the hydrogels, we also have some special plates and functional assays and a recovery solution and a viability assay. So now focusing on the MPS models, I will be presenting some data on co-culture, organoid, and a new EMT model. Starting for the co-culture, sorry. We have seen that our formulations are able to support more than one cell type culture. So in this case, I am going to present some data of fibroblasts and cancer cells culturing in one of our formulations, vitamin hydrogen matrix. And what we can see here is that cells are able to not only grow, but also adapt their normal morphology. In the case of the fibroblasts are allowed to from these networking structures between the cells. And in the case of the breast cancer cells, they are able to adopt these spheroid formations. And when we co-culture these two cell types together, what we can see is that not only these cells are able to keep maintaining this regular normal morphology, but they are also able to interact with the matrix and move inside or outside the matrix and establish a real connection, a real co-culture between these two cell types. Regarding organic culture, we do have a specific formulation. It's a Vigorel organoid. It's called Vigorel organoid. It's one of our ready-to-use versions. And as you can see, the protocol, it's really easy and straightforward. As I mentioned, the user basically just need to mix the hydrogel with the cell suspension and plate it and start the culture and wait for the organoids to grow. And this protocol can be used. It's compatible with many culture method. It's not only available for the DOM method, that it's usually the most popular for organic cultures, either 2D or 3D. But you can also use it to prepare a 3D encapsulation method, and also with the droplet method, where you can have a unique organoid within each droplet, and it's really good for scaling up cultures. And that same protocol can be used, for example, for establishing co-cultures with organoids and other cell lines, and also supports the culture of many sources. Many cell sources, either they are, if they are fragments of PDX or even stem cells, you can easily generate organoids derived from stem cells. And for any of these methods, this is the picture of the organoids. For any of these methods, you can easily assess the cell viability of the organoids with the Cyto3D. So this is our viability assay. And as you can see, the dye can fully penetrate the organoids and reach to the core. And it's really easy to identify or distinguish between alive cells and dead cells. One unique property, one unique feature that Vigorel has is that it supports apical out structure of organoids. That this apical out structure resembles more the organoid, the physiological organoid structure. So it's a more accurate model to assess, for example, drug screening assays in precision medicine. So what we can see here, these are intestinal organoids culturing Vigorel. This phalloidin, it's an actin marker, and what we can see is that the actin is outside in in the apical structure. So actin, it's an apical locator, and then in TAPI we can see the nucleus, and what we can see, these are young organisms that they maintain this apical out structure not only when they were young, but they keep it also when they mature. And they are able to also form these lumen-like structures and maintain it during culture. And when we compare these cultures with in matricial cultures, just move this here, what we can see is that when we culture intestinal organelle in matrigel, they develop these apical in structures, and they are more like a messy and more mixed structure. And this is since the actin, the microvilli are inside the organelles, this wouldn't be a really accurate model to assess, for example, drugs testing. Well, it's not moving, sorry. There. Another feature that Vitrogel allows is to maintain organelles in culture for a long time. So we have seen that these organelles, these intestinal organelles, are able to be maintaining Vitrogel for at least 30 minutes, but 60 days, potentially more time. And during this time, they always maintain their physiological and morphological structures. While in matrigel, we see that after 30 days, they start dying. And also with Vitrogel, it's really easy to expand the cultures, either to subculture them, to expand them, or to maintain them. Moving forward to the EMT model, so the epithelium to mesenchymal transition, during this process, cells lose their epithelial phenotype and they become more mesenchymal. And at this stage, cells usually are able to leave the primary site, circulate, and reach to a secondary site when they are able to metastasize and form tumors. So we have developed a model to establish this EMT transition. Again, with a really easy and straightforward protocol. Basically, we incubated these cells in ultra-low attachment plates during overnight, and at the following day we add the Vitrogel with some supplements and just culture them and wait for the tumors to develop. And then these tumors can be used either for drug testing or viability assays or anything that you want. So we monitor the tumor growth during time, and what we can see is that cells can rapidly start polarizing and protruding outside the spheroid. By day 7, we already observed this tube-like structure. So this tube-like structure is full of cells that are able to propagate really fast, and they start forming this outer shell structure. And by day 14, this tumor is already formed, it's mature. This outer shell structure is already formed and maintained during time. So to further characterize this model, we evaluated different markers as cancer stem cell markers and EMT markers. So the reason that we decided to analyze cancer stem cell markers is that we know that these stem cells are the ones that give rise to cancer progenitors. They are the ones who are going to develop the organoids. So what we can see is OCT4A is a cancer stem cell marker. And we see that this marker it's present in the tube-like structure. While vimentin, so vimentin is a mesenchymal marker, we observe vimentin mainly on the outer shell, but also when we zoom in this area here, that is the area where the cells are leaving this tube-like structure. Vimentin is also present in this area too. So what we can see here is that the cancer stem cells are the ones who give rise to this tube-like structure. And while cells start leaving the two-lap structure and forming the outer shell, they go through this epithelium to mesenchymal transition. They became epithelial, and they are highly metastatic and form this outer shell. To further analyze this model, we assess what was the viability of these cells after the tumor was already formed. So these are 2 weeks tumoroids, and we observed that cells, so green is alive, red is dead. And what we can see is that most cells are alive, both in the inner core and in the outer shell. So once we finish characterizing this model and confirm the epithelium to mesenchymal transition, we assess how this model could be used as a drug screening platform. So we used, let me just move this here. We use 3 days tumoroids and assay with EMT, a high dose of EMSAT, and analyze how tumors kept growing or not after 24 hours. And what we can see is that these spheroids, the tumors that were treated, they cannot develop this tube-like structure. So the treatment prevented this form of tube-like structure, and also have a higher presence of cells deaths in the inner core compared to the untreated tumors that are, like I mentioned, these cells in the tube-like structure are really fast propagated. So within 1 day, they really can develop this tube-like structure. So this result say that this model, it's a good model to both, on the one hand, develop these tumoroids and maintain any culture, but also use it as a drug screening platform. I cannot move there. And just so you know, Vector is able to support many other MPS models. We didn't want to give too much information, but all this information is our webpage. So if you want to, we invite you to check our website and contact us for more information. Thank you.
Thank you so much, Eugenia. That was a really great presentation. We do have about a minute for questions. So again, if there's any, I don't see any yet specifically for you, but if there's any questions for Eugenia or TheWell, please put them in the Q&A box now. Okay, there's one question that's come in. For how long can tumoroids be cultured in Vitrogel?
Okay, so we have cultured them for at least 2 weeks, at least 2 months, but potentially they can be cultured for longer. We do know that at least 2 months they are still alive.
My name is Lisa Hötting. I'm a senior scientist at InSphero, and in the next couple of minutes, I will give you an overview about our multi-organ-on-a-chip technologies. But before jumping into the topic, I will quickly give you or spend some words about InSphero. InSphero is a biotech company that was founded 16 years ago and has its headquarters in Zurich, Switzerland. We are currently over 75 employees who are working on the development of 3D in vitro models and technologies for drug efficacy testing and safety assessment. To do so, we are producing more than 30,000 spheroids per month, which we are calling microtissues. Our microtissues consist of primary cells. So here are some examples of our core areas. So we have pancreatic islet model to study islet biology, mainly focusing on type 1 and type 2 diabetes. We have a few liver models for liver safety and disease modeling, and tumor microtissues to study oncology and immunologically related questions. Besides this tissue and disease modeling, we do our own microplate engineering, including also our Acura organ-on-a-chip platforms. And we have experts specialized in endpoint technology development, from automation to transcriptomics, high-content imaging, cryopreservation, and biobanking. So, having in mind that our 3D microtissues are proven standard for efficacy and safety testing, trusted by leading scientists in the pharma, I will now talk a little bit about how our Acura organ-on-a-chip platforms built on this foundation to offer more deeper biological insight. So by using our Acura organ-on-a-chip platforms, we can model tissue-tissue interaction in vitro to better understand how do different tissues influence each other. We can add immune cells to understand how do immune cells interact with the tissue and the health or disease condition. And we have the opportunity to add physiological flow conditions to better understand how does continuous flow affect cellular behavior and drug response. So the Acura organ-on-a-chip platform is a tool that helps to better understand all these questions. Meanwhile, we have 3 different platforms, which I will go into detail in a second. They have all unique features supporting specific application, but what they all have in mind is that they can be easily integrated into your already existing 3D cell culture workflow, which means that we have an off-chip production and on-chip integration, which means that you can produce your spheroids according to your established protocol. You QC them, and when they are functional active, only then you can transfer them in the Acura organ-on-a-chip platforms. Therefore, we have established manual or semi-automated or automated transfer protocols, so depending on the support that you were needed. The crosstalk is initiated by gravity-based flow. Therefore, the organ-on-a-chip platforms were placed in this Acura all-in-one tilting unit that you can see here, which fits into an incubator, and by tilting back and forth, we can induce the flow. And last but not least, what was very important while developing all these platforms is that they are compatible with already established endpoint analysis. So all our plates are compatible with high content imaging. Then the spheroids can be removed afterwards for downstream analysis, including omic approaches or any biochemical assays. And of course, during the anti-experiments, you have access to the supernatants, not only for medium exchange, but also for analyzing of the supernatant. And with that, I would like now to start with an example application of the Acura flow system, our most scalable multi-organ system, which is actually also our first one that we had was almost developed 10 years ago. So this is how it looks like. You can see here a polystyrene chip. Each chip consists of 2 microfluidic channels, with each having 10 compartments. In each compartment, you can place a spheroid, and at both ends you have a reservoir. So now by tilting back and forth, the liquid flows from the upper reservoir along the channel, interconnecting the individual compartments, and by this inducing the crosstalk, vice versa. And then 4 of the chip fits into an handling frame, and then 4 handling frames can be placed on this tilting device. So in simple words, the Acura Flow platform can connect up to 10 spheroids in a lane. And one application where we used the Acura flow platform was to recapitulate glucose and lipid metabolism. Therefore, we have co-cultured liver and islet microtissue in the Acura flow chip, because those organs or tissues are the key regulator who are responsible for keeping our blood glucose level in a very tight range, and even little disturbances in this homeostasis lead to severe diseases like diabetes, obesity, or fatty liver. To reflect a little bit the physiological ratio in the human body, we have combined 8 liver with 2 islets in the chip, and this is how it looks like. This is an image of one microfluidic chip with the liver and islets. As mentioned before, this off-chip production, on-chip integration. So we produced the liver and the islet according to the established protocol. Then we transferred them into the chip, and then we had 2 different conditions. So we either cultured them under physiological glucose or under high glucose level remodeling disease conditions. And of course, both conditions were without insulin, because insulin is secreted by the islet. And then after 7 days of co-culture, we checked several specific endpoints. So we checked insulin secretion, which is increased under the disease state, reflecting the in vivo situation. And this directly affects also the liver-specific endpoints. So we can see that due to this increased level of insulin, we also have a high level of glycogen in the liver, as well as a higher level of triglycerides by an induction of de novo lipogenesis. And all this then leads to a dysfunctional glucose-stimulated insulin secretion of the islets. All in all, this is an example showing that we can use the Acura flow chip to recapitulate this crosstalk in vitro, which may then have the potential to discover new therapies focusing on this interplay. And with that, one last sentence. So the plate is not only exclusively for liver islet co-culture. So depending on the organ that you transfer into the chip and your experimental setup, you have, it's open for a wide range of application with very flexible endpoint analysis. And with that, I would like now to move on to our Acura twin plate. This is more a simple organ crosstalk plate where you can co-culture 2 different organs in the presence of immune cells. So here I would like to focus on one specific application that we established in-house, which is belongs to the field of immune-mediated safety and therapeutic impact. So here we worked on or established how we can simultaneously assess the therapeutic effect of bispecific antibodies and its off-target toxicity in the liver. So just briefly some background information. So bispecific antibodies have the clinical benefit that they can redirect all T cell population towards cancer cells to facilitate efficient tumor cell killing. However, this also leads to the side effect of systemic cytokine release, which can lead to liver toxicity. To recapitulate this, then in vitro, we have combined a liver and a tumor. So liver shown here in green, the tumor in red in the presence of immune cells stained in yellow. So as I said, a very simple crosstalk, but it's highly scalable. So up to 192 conditions can be cultured on one plate and analyzed in parallel. Just one example, what you can measure. So here we have co-cultured liver tumor and PBMCs in the presence of bispecific antibodies. And what you can see is that in the presence of the bispecific antibodies, we have an increase in inflammatory cytokines, leading to tumor killing over time, but also leads to the side effect of an increase in ALT release, which is a specific marker for liver damage. So indicating that we also have here this liver damage. Similar effect could be shown with another bispecific antibody, and here we could show that with co-culture, co-treatment of another antibody, that we can mitigate this ALT, so the liver toxicity, while having still an efficient tumor growth. This is how this plate looked like. So the twin plate is based on our 384 well format. So you have always two adjusted wells are interconnected with a very thin channel. So you can separate the different tissues in both wells, but they are connected by tilting back and forth via this channel. So cytokines or secreted molecules from the organs are interchanged, and also the immune cells are mixed. And last but not least, I also want to show you an example of our Acura Immune Flow. This is a platform which is more specific if you're interested in long-term immune cell perfusion, and if you're interested how individual immune cells or individual cells interact with tissue. So this is how the chip, it's again a chip format where you have here the compartments where you can co-culture the spheroids. And then you have the single cells, for example, cancer cells, circulating cancer cells, immune cells in the medium, and they pass by the spheroid, and then from time to time they attach. So you really have this single cell tissue interaction. And this feature was very important for one application that was performed by our cooperation partner, Dr. Franziska Linke at the Erasmus Medical Center in Rotterdam. So she assessed the cancer potential in the chip. Therefore, she has co-cultured liver and bone spheroid in the chip. And then she added prostate cancer cell line into it, as you can see here, the experimental setup, so combining bone and liver microtissues. And then she really could see after 3 to 4 days how individual prostate cancer cell attached to the liver and the bone. And then after 3 to 4 days, the circulating prostate cancer cells were removed, and then she monitored only the attached prostate cancer cells, how they attach and proliferate or invade into the tissue. So this is work on progress. So here just monitoring and detecting the frequency on microtissues which were attached by the prostate cancer cells. And there you can really see nicely differences between the liver and the bone, reflecting how this cell line behave in vivo, which is a clear difference to how it is in static condition, where you always have 100% frequency. And yeah, with that, I'm coming to the end. So I hope I could show you that our Acura organ-on-a-chip platforms can augment your 3D in vitro assay by deepen your biological insight while simultaneously preserving the scalability, having low operation complexity for reproducible and reliable results, and that all the platforms can be compatible with all your existing endpoints in the lab, so that you don't need to invent everything from scratch and that you have a smooth integration in your workflow. And with that, I would like to thank you, my colleagues, and our cooperation partners.
All speakers come back on camera, and we will give everyone a few minutes to think of their questions. I know there was a few individual company questions in the Q&A, but I think Mornay and Muyil, you both answered those already. So we'll just give everyone a few minutes, and maybe in the meantime, I can start off with a question for all the companies, so we can just go one by one. So I know obviously drug discovery versus development is a very relevant topic. So I'm wondering, from a broad perspective, do you see your multi-organ models that you've talked about today more applicable in drug discovery, development, or both? And why? So maybe we can start off, we can just kind of go in order from presenters. So Mornay, if you want to kick us off with that question.
Sure, thanks for the question. I think our gut liver model probably fits in better in lead optimization. So probably once you have a couple of drug candidates and you're trying to maybe optimize on them, determining a bioavailability, I see it's fitting in that step before going on to first in-human trials. So you can get an idea of the parameters such as permeability, clearance, and bioavailability.
Great, thank you. Muyil, what about your models?
Yeah, so our pillar perfusion plates are designed for high throughput screening of organ toxicities and drug efficacy. So our platforms are not specifically designed for one individual organ system. It can be applicable to broad cell types. We can use a human organoid. We can use a cancer organoid. Also, we can incorporate immune cells or other cell types in the perfusion plate. So it's very, very versatile system.
Great, thank you, Muyil. Eugenia.
Yeah, thank you. In our case, yes, VitalDrug could be used for both, either for development and drug testing. In the case of the organoids, we have seen that VitalDrug organoid is able to support many organoid types, not only intestine, but also pancreatic, lung, prostate. And since they are able to develop to support this apical structure, it's a really good and accurate model to do drug screening assays.
Yeah, thank you, Eugenia.
Yeah.
And Lisa.
So I would say maybe I need to discriminate a little bit, because we have at InSphero a lot of already established assays for safety assessment and drug efficacy testing in the early and later state. But I would say the models that I presented here today is more the explorative area, so also more for lead optimization, or in general just for fundamental biology tools to understand the process itself. So
Great.
And of course, with the twin plate, it's I think there we have a very mature system, which can also be through throughput applied for industry settings.
Great. Yeah, thank you, Lisa. I have another question that I can ask, kind of again for all companies. So obviously with the 3 Rs being more and more top of mind, Can each of you talk a little bit about how your multi-organ models forward the 3 R's of animal research? So either through replacing animal studies, improving their design, or refining animal pain and distress. And if you have them available, are you able to speak to any animal numbers replaced, potentially by your models? So we can just go in the same order. Mornay, if you want to start.
Sure. So I think our system probably fits more in the reducing and replacing animal testing. It's a bit too early now, so I can't tell you how many animal studies we've replaced, but it definitely fits in that part of the pipeline where you can, rather, instead of getting values from animal studies like we showed in my talk, that there isn't very good correlation between a lot of the animal studies and human in vivo observations. So we really see our setup really fitting at that part in the pipeline where you can rather get a more accurate prediction of your parameters for your drug from primary human cells and then take that forward. Yeah.
Great. Thank you, Mornay. Muyil.
Yes, so our system can be used for reducing animal studies. So for example, I showed 2 examples today. One is the liver regeneration, which is typically done in animals. Our organoids are regenerative, so which means that you can treat the organoid with the model compounds and then incubate it in the expansion media so that you can check and see if your liver can truly regenerate it after drug treatment. That's one example. And our system actually models blood flow in the microcapillaries. Unlike other systems, we can generate extremely fast flow rate in the microfluidic device so that we can truly mimic the flows of immune cells in the bloodstream. And then we can also separately simulate the tumor microenvironment on the pillar plate. So we can incorporate the ECMs and other cell types in the tumor microenvironment on the pillar plate. So that's another examples of reducing the animal models. And finally, our brain organoid has been used for testing the developmental neurotoxicities, which is currently done with the animal models. So we are aiming for these animal reductions. I don't have specific numbers though.
That's okay, no problem. Thank you, Muyil. Eugenia.
Yeah, well, we all know that for Madrigal, for example, it takes up to 7 mice to develop just 1 vial. So anything that is synthetic, like our hydrogel, is going to reduce a lot the use of animals. And also, since it's room temperature stable, we don't need to use any dry size on the shipping. So we also, with our product, using our product, moves to reducing the carbon footprint. And regarding the stop or replacing the use of animals, as we showed, our system is really able to reproduce the microenvironment for cells, not only for organoids or for tumors, but depending on the aim of the projects, whether you're going to be doing a 2D or 3D culture, or organoids or tumors, or even scaling up or high throughput applications. Our hydrogel is really easy to use and really mimics and reproduces the microenvironment. So it really moves to stop using animal-derived products or animal-derived cells, and being able to easily culture human-derived cells, for example, or human-derived samples. Like we do have some really good and pretty new application for maintaining human tissue, breast cancer tissue, to be able to use directly the patient's tissue to assess personalized medicine and assess specific drugs that will be able to attack these particular carcinoid in one particular patient.
Awesome. Really interesting. Thanks, Eugenia.
Yeah.
And Lisa.
So I would say that our technology falls into the category to, on the one hand, to reduce animal experiment by sorting out earlier non-effective compounds and sort for the ones that are non-toxic. So to increase actually the good drug candidates. And on the other hand, so our assays can be used maybe even to replace them. So the data that are done with our assays can be used for IND applications.
Awesome. Thank you so much, everyone. I'm not seeing any new questions come through the Q&A. So I think at this time then we'll go ahead and end the meeting.